All right, everyone. So today what we're going to be looking at is the process of cellular respiration. So in this PowerPoint presentation thing that I'm going to talk about, we're going to go over the molecular process of what's happening inside of the cell. So when we're thinking of respiration, right, we're often thinking about breathing and taking in air and then releasing that air and that oxygen that we take in and releasing it as carbon dioxide into the environment around us. But the process that we're going to focus on today is look at cellular respiration on the cellular level. So what are the actual chemical reactions that are happening inside of your cell for us to take the oxygen in, to break down the food that we consume, to generate the energy we need in order to carry out all of our cellular processes. Okay. So let's go ahead and start talking about that. So there are a few things that we want to think about as we're going through this whole process. So we've talked about how energy and action are linked, right? So the idea that all of the energy that we have is converted into some kind of mechanical force, right? If we push or pull something like that, then we're actually generating that particular force, right, in order to make something happen, right? We need that energy to do work. Okay, so then the question is, how can we take that stored chemical potential energy that's inside of all of these macromolecules, like your proteins, lipids, and carbohydrates, and convert them into some form that's actually usable? Like, how do I take those sugars to like lift something, like this bag of VGA cable, right? So what energy is necessary to make that happen? So in this lecture, we're going to focus on the cellular level, right? Our basic unit of life, right? And how we get the energy necessary that we can then scale up to get these larger forces to occur. Okay, so cellular respiration. Here you guys have probably seen this basic formula, before, right, for cellular respiration. Okay, so in this formula, what you're looking at is you've got your chemical reaction here. Okay, so we're starting with some kind of glucose molecule, so C6H12O6, and in that process, we're converting that energy, right, or we're converting that stored energy with oxygen gas, so using oxygen as a means for our reaction, right, as part of our reactant, to produce carbon dioxide and water. So this overall reaction doesn't necessarily highlight any of the major steps that are happening, right? Because we're only taking this molecule and breaking it down into constituent components. But it's important to know this particular formula because this formula can be scaled up and we can use it once we talk about all the things we learned today, all right, to figure out exactly like how many molecules of energy are we producing, or storage molecules are we producing that can carry out all the processes inside of our cells. All right, so with your cellular process, it's important to remember that this process is a catabolic pathway. Okay, so what that means, as if we're talking about anything that is a pathway is going to be a series of reactions that happen in order to get a final product. Okay, so along that pathway, things can then spread and go into other directions. You can get other things that happen. But for our purpose, when we're talking about this catabolic pathway, we're talking about all of the different reactions that use oxygen, right, to break down this glucose molecule, right, the C6H12O6, into its smaller constituent components, right? So we're talking about this molecule here and breaking it into other things. Okay, so one big part of this to remember is that oxygen is always present when we're doing cellular respiration for a large portion of the reaction process. Okay, so oxygen is consumed as we break down organic molecules into its basic form so that we can then use it for energy. So this includes carbohydrates, lipids, and proteins, any food. But what we're going to really focus on today is we're really going to talk about and look at and discuss. This camera's bothering me. And discuss what's happening with glucose, right? That's our most basic, most common form of energy that we utilize as humans in order to get the ATP that we need, or the molecule that we need that actually carries out all of the force and reactions later. Okay, so there are three main stages that you're going to be responsible for knowing, okay? And that is glycolysis, the Krebs cycle, or also known as the citric acid cycle. Some of the images call it that, and I'll highlight that a little bit later, and then the electron transport chain. So these three processes in the pathway of cellular respiration lead to the generation of the energy molecules that we need to carry out the reactions inside of our cells. Okay. All right, so this diagram right here shows you an overview of the cellular processes that happen inside of your cell. Okay, so just to break down this image and look at a few things, there's different places that this stuff happens. Okay, so you guys might remember this term cytoplasm. So cytoplasm is referring to just the inside of your cell. Okay, the other structure that's important for cellular respiration is the mitochondrion. So I'm sure you all have heard the mitochondria is the powerhouse of the cell, right? And that's because the mitochondria is the place where we are generating these molecules that carry out all of the other functions. It is the powerhouse, and that it is the location for where these things happen. Okay? So if we look here, our first step, glycolysis, happens in the cytosol, and we'll talk about that a little bit. Then you have your Krebs cycle and your electron transport chain that happen within the mitochondria. And we'll talk more specifically about where those happen. So first, right, we've been talking about energy for a long time now, but when we're talking about energy inside of a cell, what we're really talking about is the energy that's stored inside of a particular molecule. So we start with our high energy molecule like glucose that contains lots of carbon bonds, right, that form a circular ring, right? We have to take those bonds and break them because the energy that's in that glucose is not available to us until those bonds are broken. Then we can use that energy to help carry out other reactions to get molecules that are more easy for our bodies to accept or to utilize to get the energy that we need. Okay, so the molecule that we use, the molecule you'll hear a lot about, is called adenosine triphosphate. So here is a little GIF of what's happening here of that process, where you have your adenosine nitrogen group with your ribose sugar and then 3 phosphate groups, so PO4s. So what's happening here is this nucleotide, so this is a nucleotide. If you think back to your macromolecules, this is a nucleotide, right? And you have 3 high energy bonds that happen. So inside of this ATP. So by creating a bond, you're storing energy, and then by breaking that bond, you can then release energy. Okay? So cells, right, use this energy to carry out pretty much all the cellular processes. We need ATP, and that could be to change the shape of an enzyme, to change the shape of a protein, to help generate something else, some process, in order to get the materials that we need. Okay. So energy is released as these bonds are broken. So this can be through some kind of enzymatic action, like an enzyme coming in, clamping onto that phosphate group, splitting it, and getting the energy out of it, or other means. There's other things that happen inside of your cells to make adenosine diphosphate. So this kind of intermediate molecule where there's only 2 ATPs. All right, so let's look at ATP chemically, again, much more zoomed in. So I want you to be able to recognize this molecule if I gave you the molecular structure of it. Okay, so if I gave you this. So in this, since it's a nucleotide, nucleotides, remember, always have 3 components, okay? You have your phosphate group, your ribose sugar, and then your nucleotide base. So in this case, it's adenine as our base. So you've got these 2 rings here that form this adenine structure, okay? So we know it's adenosine triphosphate because we have these 3 phosphate groups that are on here, okay? So if we take one away and break this bond, then we're gonna release energy so that you can then, wow, whoa, what's happening here? You can release energy, right? So that then you can carry that energy off into some, or use that energy for something else, okay? All right, there are other molecules that are important for us to track in this entire process. So as cellular respiration happens, we're breaking a lot of bonds. So by breaking bonds, we're releasing other things. So hydrogen ions are released into the cytoplasm or into the matrix inside of your mitochondria. You've got electrons that are released that are then stored by other materials. So 2 molecules that are important for us to keep track of are NADH and FADH2. So these are both large molecules that contain, a bunch of different places, but mainly what's happening here with these and what you need to know is that they are electron storage molecules. What they do is they take electrons that are released in the cellular respiration process and store them for later. And so they use those electrons as energy later in our last process of the step of our electron transport chain. Okay. So FAD does the same thing, NAD does the same thing, and we'll see these similar molecules when we talk about photosynthesis. They're a little bit different, but close. Okay, but your important thing to remember here is that NAD and FAD are both electron acceptors. Okay. All right, so where does this whole thing go down? So I really like this diagram because it shows and highlights where you're going to find and look at all of these different reactions that are occurring. Okay. All right, so if we're talking about what's happening in our cellular respiration process, okay, all right, it actually takes place in 2 parts of the cell. Okay, so the first part that happens in the cytosol, right? Cytosol is just another term, or you might think of it as the cytoplasm, right? It's happening within the cell membrane, within the liquidy structure that makes up the cell, okay? All right, the second part of this reaction, so the citric acid cycle, also known as the Krebs cycle, okay, so both of those are the same thing, right? Citric acid cycle or Krebs cycle happens inside of the mitochondria as well as the electron transport chain. Okay, so both of those things occur within these in the mitochondria. They happen in two different places inside of the mitochondria. The citric acid cycle happens within this space, kind of in here, with inside of the folds, called the matrix. And then our electron transport chain happens in the actual membrane folds inside of the mitochondria. Okay, but remember, glycolysis, cytosol, everything else, mitochondria. Right. So if we're looking at the mitochondria, well, that was fancy. If we look at the mitochondria structurally, just to recall, right, to look at the structure overall, right, you have this internal area here, right, the outer, that's known as the matrix, okay. You have the cristae, which are these folds of this inner membrane, and then you have the outer membrane around the mitochondria, okay. So the folds are called the cristae, and the matrix is called the inner portion, the inner fluid portion, where some of our reactions occur. Okay, so it's important for you to remember those. It's also important to remember that you have these membranes, right? And what we know about membranes is that on either side you're going to have varying concentrations of things. So for example, you might have a whole bunch of hydrogen ions on the outside of here, right, that want to move into this area, right? And so that movement is something that we'll address a little bit later because it's important for actually generating ATP. All right, so here's a zoom in version of that image. All right, so I would recommend looking at this, memorizing this, having an idea of what's going on in these major places is going to be very useful for you. All right, so let's talk about each of our individual processes. So let's start with glycolysis. Right? So glycolysis, right, if you take that term glycolysis and break it down to its two constituent points, glyco refers to sugar, right, and lysis means to split. So you're literally splitting sugar when you're doing this, right? You're taking that C6H12O6 and producing smaller versions of sugars from it. Okay? All right, so this occurs within the cytosol of our cell, right? And what happens here is you take that glucose molecule, that C6H12O6, and break it down into two smaller molecules, into two what we call pyruvates, or you might see pyruvic acid as another molecule. And what's happening here is you are just chemically cleaving that sugar, that circular ring of carbons, into two smaller 3-carbon chains. Okay. All right, this whole process actually occurs without oxygen. So this is our anaerobic process that begins cellular respiration. So you might be asking, well, okay, if I don't need oxygen to make this happen, why does it matter? The problem is that glycolysis is not very efficient, right? You don't get very much energy from breaking a single glucose, right? It takes a lot more, it's not really available. It takes more chemical reactions to happen in order for us to get as much energy as we need to do anything, right? Me lifting up this like little key thing, right, is going to take way more energy than I can get from glycolysis. All right, the other thing to remember this is that it's an exergonic reaction, or some of you might think of it as an exothermic reaction, and that you are releasing energy as you do this. So most of that energy is not lost, right? We end up taking that energy and converting it to another form, partially by taking our electrons and storing it with NADH, and also by generating some ATP, right? You actually generate some new ATP molecules from ADP in this process. Okay, so here's a diagrammatic view of the entire process. So what we're doing here is we're actually inputting 2 ATP molecules and a glucose, right, in glycolysis in the cytosol. It's happening in the cytosol. And our output is going to be 4 ATP and 2 NADHs. Okay, so you're going to get, or 4, no, it should be 2. Sorry, it should be 2 ATPs. So you get 2 ATPs and 2 NADHs as you're going through this process for pyruvic acid. Okay, so you're going to get 2 of these. So pyruvic acid is the same thing as a pyruvate. Got a new like typey thing, and it's weird. I'm getting used to it. Okay. All right, so here if you're looking at the entire process, this is FYI, I don't expect you to memorize this, but this is what we're doing. We're taking that glucose, right? We're splitting the circular form of that glucose molecule into a 6-carbon chain. We're adding some different things to it. We're taking some stuff away, and we're generating, or yeah, we are generating 4 ATP. Yeah, 4 ATP per glucose. That's where I'm getting confused. All right, so as you're going through this entire process, you are adding phosphate groups, right? Those phosphates can then be used for other reactions. You're splitting the molecule in half, and basically in the end, you're getting 2 pyruvic acids at the ends, or 2 pyruvates at the ends. Okay. So after you go through glycolysis, there is a step that happens before we reach the Krebs cycle. So I didn't really highlight that as a big thing, but we need to somehow get that glucose that's been broken down into pyruvate into our mitochondria. So that only happens if we're able to transfer it from the cytosol into our actual mitochondria. Okay. And this junction occurs between glycolysis and the Krebs cycle, or the citric acid cycle. Okay. And what happens is we actually use oxygen to oxidate pyruvate and add what's on add onto it a coenzyme. So this coenzyme essentially takes that molecule, breaks it into a smaller piece, right? We get a little bit of energy from it, right, in the form of NADH, and we generate our first carbon dioxide molecule. So this is where we're starting to get those products of the chemical reaction, that carbon dioxide. Okay, so in this process, we're going from a 3-carbon pyruvate to a 2-carbon sugar, right? And then we're attaching coenzyme A to it. So this acts as a carrier molecule. It's a signal molecule that when the mitochondrial membrane sees it, they're like, oh, come on in, right? So just to look at this, right, for this entire process, you've got your pyruvic acid here, right? You're generating CO2 as you input a coenzyme onto this molecule, and you're also releasing NADH in this hydrogen ion. So one thing I don't necessarily highlight in the text in this PowerPoint is the importance of this hydrogen ions. We need to keep track of these. These hydrogen ions are really important later when we talk about the electron transport chain. Okay, so this is just to give you an idea of where that's happening. So we have glycolysis and we have form our acetyl-CoA. So this allows it to then transfer into our mitochondria so we can start going in through the Krebs cycle. So we can end up hitting this particular process. All right, so the Krebs cycle. So the Krebs cycle as a reaction is long. There are a lot of steps, there are a lot of things that are happening in this process, and you can thank this guy Krebs for actually figuring this out. You guys are not going to be responsible for knowing all the steps of the Krebs cycle. I'm going to show you kind of an intermediate version of what it looks like, and also a version that I expect you to be able to reproduce. Okay, so things about the Krebs cycle to remember. 1, this requires oxygen to happen. This is an aerobic reaction. So anything that requires oxygen, we say it's aerobic. Okay, so this is a cyclical process. So it's actually a continual ring, right? We call it a cycle for a reason. It's because it circles around. And as we go through this, we're only producing 1 ATP per cycle, but we are producing a lot of those high energy NADH and FADH2 molecules that we can use for future reactions. Okay, so per glucose molecule, they actually have 2 cycles that occur in the Krebs cycle. So I want you to know that. You get 2 ATP out of it, and all of this takes place within the matrix of the mitochondria. So if we go back to the structure of the mitochondria, right, it's going to be this inner portion here, right? This is where our matrix is. That's where our citric acid reaction is occurring. Okay. All right, so this is the image of the Krebs cycle that you should know. Okay, you should learn this. All right, so what you're starting with is your acetyl-CoA. All right, so you've got your 2-carbon sugar with that coenzyme A. Coenzyme A comes in, it gets released through one reaction. And in that process, you're taking those carbons and splitting the bonds and storing some of that energy into NADH. Okay, so in that process, you're actually releasing 2 carbon dioxide. So you're actually getting rid of some of that carbon in the form of carbon dioxide in the process of oxidizing it or reacting it with oxygen. So anytime you take sugar and oxidize it, you produce carbon dioxide. Okay. So in this process, you're also generating an ADP that gets changed to an ATP, and you're getting FADH2. So again, you're getting 3 NADHs and FADH, and I'll talk about that here in a second. But you should be able to reciprocate or produce this particular process. Okay. So this right here is the much more elaborate version of it. So in the process, you actually generate first, when you have this coenzyme A come in, you actually do produce citric acid. That's why it's called the citric acid cycle. We use that term, we use both those terms when we're talking about Krebs cycle to name it after the individual who discovered it, and citric acid cycle because that's what you're generating. Okay, but again, all of these reactions occur. I think it's important for you guys to see how complicated this actual process is. All right, and know that we are having a lot of different chemical reactions happen, all right, in this catabolic reaction. Okay, but you just need to know that we're producing NADH, FADH2, and we're getting a little bit, 1 ATP out of each of these different terms. Right. So just to summarize, right, these are the things that I want you to be able to walk away with and know, right, is the products of this particular process. Okay, so each turn produces 3 NADHs, 1 FADH, and 2 carbon dioxides. So that's our kind of 3 carbon dioxides that we produce per turn. Okay, so if we go through a whole process for each of our glucoses, right, that's our 6 CO2s that we're producing. All right, so therefore, for each glucose molecule in the Krebs cycle, you're making 6 NADHs, 2 FADHs, 4 CO2s in the Krebs cycle, plus the 2 that you get from glycolysis, and 2 ATPs. Okay, so the big takeaways here is you're producing these molecules here that are important for our electron transport chain, our last reaction. We're getting our waste product from the reaction, and we're generating 2 ATPs. So again, you're probably thinking like, Joey, there's not a lot happening here. Like I've only made 4 ATPs so far. And that's true. Glycolysis and Krebs cycle are not very efficient in terms of generating ATP, right? There are other ways that we use the energy that comes from that glucose in our next reaction. Okay. All right, so we're up through here. We've talked about glycolysis making our pyruvic acid. Then we got our Krebs cycle happening where we're making some ATP and producing our CO2 byproducts. Our last thing that's happening is our electron transport chain and chemiosmosis. I can never say that word. But this is where we're going to be making the most ATP. The most ATP comes from this last part of the reaction. So we've done all these steps, we've built up all this energy, we have all of these gradients that have formed, right, with all of these hydrogen ions we made. All right, now we're going to get to the big picture where we're going to make all of this energy. Okay, there's a lot of text on this slide, I'm sorry, but it's a good summary of what's everything's happening. At least I think it is because I mean this. All right, so electron transport chain. So this is our last step in the entire process of cellular respiration. All right, so this is actually happening within the inner membrane of our mitochondria. So not on the outer membrane that surrounds it, but actually inside of those cristae folds in our mitochondria. So what ends up happening is oxygen gets pulled into this area, right, and it actually is used then as a reactant to pull down electrons from the NADH and FADH2. So remember, oxygen has a high electronegativity, right? It really wants those electrons in order to make itself happy, right, to be electrically stable. Okay. Right. So it's going to pull down those electrons from those different things, and in that process, it actually makes the electrons move down a chain, right? We call it the electron transport chain, through a series of enzymatic reactions on a whole bunch of different proteins that occur in that membrane, right? You guys are not responsible for knowing all of those proteins, but you do need to know that in this process, right, electrons are going from a high energy stored state in the NADH to actually reacting with oxygen gas to be released as a product. Okay. All right, our process here is going to generate about 90% of the ATP your body needs. So in general, it makes about 34 ATP. So that 34 is, it's like, yeah, 34 if the reaction occurs at 100% perfect rate. As we've seen, as we've gone through some of our stoichiometric reactions, or if you've ever done stoichiometry before and you found a limiting reagent, right, sometimes your reactions don't necessarily go complete, right? You don't fully react every single part of it. So you don't get 100% efficiency. And that's true for your biological systems. They don't always work perfectly. Okay. So again, you need oxygen as your final electron receptor. So you're actually taking oxygen and using that to store electrons that then get released, okay, and used for other things, right? So remember, for every FAD2 you make, you get 2 ATPs, and then for every NADH you make, you get 3 ATPs. Okay. So then our whole process here is coming down to chemiosmosis, where we're actually, if you remember, osmosis is the movement of water. So chemiosmosis is actually the movement of hydrogen ions across a membrane, specifically through a protein which is known as ATP synthase. Okay, so this protein right here is probably the most important biological protein in any system. Okay. All right, ATP synthase is the molecule that allows you to actually stick these phosphates onto ADP. Okay, and it does it really, really quickly, and it does it in a very cool way. It actually rotates. It's sort of like a rotor. And what's happening is you're actually taking this like molecule that's coming in and you're like jamming on this phosphate group to it, then releasing it out into the world. Okay, so it's like a spinning cycle that's like slowly clicking it on. It's pretty cool. All right, so ATP synthase, right, again, is in the inner membrane of the mitochondria, right, and it uses energy that's from this ion gradient. So remember, we've been making a whole bunch of hydrogen ions as we've been releasing these electrons and storing them in NADH and FADH2, right? So anytime you lose those hydrogens, right, and electrons, right, you're just left with a single proton. Remember, a hydrogen ion like this is equal to a proton, right? So that proton, right, ends up being on one side of the membrane. So you have a whole bunch of hydrogens up here. I'm still learning how to use this really cool little writer thing. All right, and then it's going to move through this enzyme where we have a lower concentration. So remember, our concentrations go from high to low. High to low. Okay, when you go from high to low. So these hydrogen ions are going to be at a very high concentration up here, low concentration down here. So they're going to move across that particular enzyme to even out that process. And as that high energy ion moves through it, it actually causes the ATP synthase to start to spin, right? As it reacts with that hydrogen, it changes the shape to get our ATP. Okay, so here is a diagrammatic view of the entire process of the electron transport chain. So again, you have a series of protein complexes that go through here, and as you have these protein complexes go through, right, your oxygen is going to end up reacting with the electrons, right, pulling hydrogens in, right, and actually generating water in the process. So this is where we produce that water as our byproduct of cellular respiration, is through the electron transport chain. Okay, so then lastly, as these electrons move through, right, and these NADHs react and lose their electrons, they're actually pumping hydrogen, right, from inside of the matrix out into the interstitial membrane, right, the space between the outer membrane and the inner membrane. So suddenly you have all of these hydrogen ions that are pumped out here, right? The gradient's going to want to go back. It doesn't want to stay uneven. So that energy will then go into through ATP synthase, right, and produce this ATP that we need. Okay, so here's a summary of all of the processes and the products that you're going to get. All right, so you start with glycolysis, you make 2 ATPs, and you produce acetyl-CoA. Okay, acetyl-CoA will, and in this process you're making NADH. Right, in this process you generate 2 NADHs, right, from making acetyl-CoA, and that acetyl-CoA then enters the citric acid or Krebs cycle. In the Krebs cycle, you're going to have NADH that's going to react here. Okay, and then our NADH is going to be produced, FADH is produced, as well as 2 ATPs. So again, so far we have 4 ATPs. All right, and then lastly, we end this with 34 ATPs being produced from our electron transport chain. So just remember, this whole process takes about 2 ATP to start. Right, so you're getting about 38 ATP, so you're netting approximately 36 ATP per cycle of this, per molecule of glucose. All right, so again, here's a written summary of all that. So if you want to pause this and write this down, that would be a good idea, right? Because you're responsible for knowing the products, right? If I say like glycolysis happened, I want you to tell me what am I making? What are the products of that particular process? If I'm saying Krebs cycle, what am I making in my as part of my product? Okay, all right, so I didn't summarize in this part what's happening in the electron transport chain. What's important for you to know is that for every NADH, I'm making 3 ATPs, right? And all of these NADHs and FADH2s, where I make only 2 for NADH, are produced through the citric acid cycle to produce this in the electron transport chain. So again, our grand total is 36 ATP net because we do utilize some ATP to make glycolysis happen. So this is assuming that we have a complete and fully efficient reaction. So not every single glucose will produce 36 ATP, okay, but we are going to assume an ideal reaction in this case for our learning process. Okay. All right, so just some things to think about as I kind of summarize all of this information I've spewed at you in this video is note that it's important to track all of the products of our cellular respiration, right? What we're actually making as our steps go on. Another thing to be thinking about is we're going to be addressing kind of the sister reaction to cellular respiration, which is photosynthesis. Like that is chemically the opposite of this process. But we'll dive more into the actual process and talk about the photosystems and things that are there. Right. Next, how do the cellular processes translate to the bigger picture, right? What does it mean as I generate this glucose to actually produce the final product, right? What does it mean for me to actually get this to happen? Okay. All right, and so your challenge that I asked for you guys to try, knowing the chemical reaction and knowing that you make 34 ATP per molecule of glucose, right? I want you to calculate how many grams or how much energy, how many ATPs you can actually make from an entire gram of glucose. So by saying like a gram, right, we're talking about like you adding like not even very much sugar to your coffee, right? It's like basically just like a sprinkling of sugar to your coffee. Okay, so I want you to think like, oh, okay, how much energy am I actually producing, right? Just to get an idea of the relative amount. Okay. All right, so again, I would recommend making sure you go through and track the products of each of the steps, that you know the order of the things that are happening, and we will talk about it more later. All right, enjoy.